again, thanks everyone for logging in. So this week it's Dr. Nako Nakatsuka, so I really hope I pronounced her name correct. And I'm very excited about this talk this week. So this is actually the first presentation of someone who I don't actually know personally in real life. So this is kind of a talk that's been generated out of the community here, we could say, in the webinar series. So I'm really excited. This is a topic close to my heart as well, on nanopipette and modifying nanopipettes for sensing. So really looking forward to it, and over to you. And thanks for giving the presentation.
Thank you so much, Mihail, and it's really a great opportunity to be here. I'm really happy that I get this opportunity, especially during Corona times, to give a talk online. And I hope that you all enjoy this topic. I must say that I'll talk about other things other than Aptamer-Modified Nanopipettes. So sorry, Mihail, if that was what you wanted to hear. But since I'm quite new to the electrochemical community, I wanted to start out by very briefly introducing myself. And so I grew up in Tokyo, Japan, and I was there until the end of my high school years. But then I moved to the US for university. I went to Fordham University in the Bronx, New York. And that was actually the first time I really got into research. I worked with Professor Ipsita Banerjee, and she was someone that really got me into this realm of research that I never knew before. And she really knew how to get me into it because I did a lot of sports growing up. I ran cross country and track and field for Fordham, and I was consistently getting injured. I was getting torn ligaments, broken bones, and my research was based on tissue engineering during my bachelor's. And it was really exciting to be able to see research that was relevant to things that I've experienced, and that was something really close to my heart. But then for my PhD, I moved across the country. I went to the University of California, Los Angeles, where I worked with Professor Ann Andrews and Paul Weiss. And so this was a slight change in my research career because then I started working and specializing in surface chemistry and designing platforms to be able to screen for DNA sequences called Aptamers, which I'll talk about in more detail later. And also that was when I entered into the realm of biosensing for the first time during my PhD. And what was really exciting was that it was really at the intersection of different disciplines. Paul is a nanoscientist and also a surface chemist, and Anne is a neuroscientist in psychiatry and neurodevelopment. And so it was really cool to be at that intersection to develop new tools that can really benefit the neuroscience community. And so now I think everybody knows from the email that you got that I'm from ETH Zurich. I've been a postdoctoral fellow from 2018 to 2020 in the Laboratory of Biosensors and Bioelectronics. This is led by Professor Janos Borosh. And here I was, thanks to the ETH postdoc fellowship, I was able to spend 2 years continuing on in the research directions of brain science, which I'll talk about today. And currently I'm still here as a senior scientist for, I believe, at least another 2 years. And so today I'll be talking a little bit about my PhD work and also the work that has been continuing on in my postdoc. And the general theme of everything is the really grand challenge of understanding the brain. And so, of course, this is a very daunting challenge. And if we take a look at the highly complicated brain, I really like to actually make this analogy of how many neurons we have in the brain. It's approximately 100 billion, which is equal to almost equal to the number of stars in our galaxy in the Milky Way. And what's really crazy is that it's not just the sheer number that is really overwhelming. It's that each of these neurons or brain cells communicate with each other. They make hundreds and thousands of connections. They communicate at the synapses. There's trillions of them in the brain. And so not only do we have so many of them, they all communicate with each other. And so if I show you a very simplified diagram of how the brain communicates, it's through two different mechanisms. First, an electrical impulse from one brain cell or neuron triggers the release of these chemicals called neurotransmitters into the synapse, which is the extracellular space in between the two neurons. Then these neurotransmitters are captured by receptors on the second neuron, and then this transduces the next electrical response. And so as you can see, it's through electrical and chemical interactions that the brain communicates. And I want you to keep this in mind because I think it's one of the key important points that I want to make for the application of our work. because there are ways to monitor neuronal electrical activity, and this is very well established. For example, in our group at the Laboratory of Biosensors and Bioelectronics, we actually specialize in bottom-up neuroscience, where in our group, we have teams that are working on patterning neuronal networks and precise patterns and morphologies on the surface of microelectrode arrays. And so these are metal electrodes that you can then stimulate these neurons electrically, or you can also stimulate and record from them and get their electronic signals. So this is in vitro. But of course, there's also in vivo recordings in the brain. And as you can see here, this is an in vivo microelectrode array that you can actually implant. It's biocompatible, it's flexible. And so a lot of research has been done in order to record electrical signals. Further, to stay with the times, I wanted to talk about NEURALINK, where of course this is the brain-machine interface that Elon Musk has been building. And if you take a look at their website, what I found was really interesting is that they say neurons communicate through electrical signals with this fancy diagram, and that we can record electrical signals in the brain. However, they really even have a diagram right here of the neurochemicals that are being also part of this mechanism, but it's completely ignored. I think what they say in terms of being able to achieve therapies for example, brain spinal injuries that are stimulated by electronics, I think that's very possible. But to make claims such as the potential to treat a wide range of neurological disorders, I think to do that, it's really important to also take into account the neurochemistry. And so there are methods that exist to measure neurochemical activity in vivo. One of them that is the gold standard is microdialysis coupled to high-performance liquid chromatography or mass spectrometry. And so in this case, you can take a sample from a specific brain region, a tissue sample, and then through a semi-permeable membrane, it actually takes out the small molecules of interest, specifically neurotransmitters, and then it goes through this separation technique, and then also the analysis with mass spectrometry. And so while this is a very accurate technique, the issue is that you can't have the spatial and temporal resolution at which these interactions are happening in the brain. And also, as you can see from this setup, it requires a lot of expensive and bulky instrumentation and expertise in order to do the measurements. There's also voltammetric techniques that really achieve the temporal resolution at which these neurotransmitters are released in the brain. And in this case, for fast scan cyclic voltammetry, you have a carbon fiber microelectrode that can then be implanted into localized brain regions in a rat, for example. And what this technique is based off of is the redox chemistry at the surface of these microelectrodes. And so we take a neurochemical, and then if you see the electrons that are being reduced in that process, then you can see that chemical in the voltammograms. However, because there are a lot of neurochemicals that are similar in structure in the brain, the issue is that they have a lot of overlapping redox potentials, so there's a selectivity issue. Also, since these carbon fibers are implanted directly in the brain, they do suffer from fouling over time as well, and also the interfering molecules start coating the surface and you get a decrease in sensitivity. And so what I wanted to show you here is this grand challenge of differentiating similarly structured neurochemicals. This is the biosynthetic pathway for two very important neurotransmitters, dopamine and serotonin. And as you can see, you have these precursors, for example, L-tyrosine and L-dopa for dopamine, L-tryptophan and L5-hydroxytryptophan for serotonin, and they all look very similar in structure. You can see that there's one extra carboxylic acid group for the L-dopa versus dopamine. Then this can be metabolized into DOPAC. And the key is we have to be able to differentiate these with very high selectivity. And the voltammetric technique suffers in this regard. And also there are antibodies for these different small molecules, but some of them are cross-reactive. And so during my PhD, the molecular recognition element that we targeted were called DNA aptamers. So these are single-stranded DNA sequences that have been isolated with the specific goal of capturing a target of interest. But what's special about the aptamers that I worked with during my PhD is that they're conformationally changing aptamers. So what that means is that in the presence of the target of interest, the aptamer undergoes a conformational change. It realigns that negatively charged backbone in order to alter its three-dimensional structure. And what is more, we believe the selectivity really comes from the fact that in the presence of the non-specific molecule, these aptamers don't undergo that conformational change. And so this inherent selectivity of the recognition element itself is really advantageous for our biosensing applications. And so I wanted to tell you two stories. The first one being the in vivo sensing application with Aptamer-Functionalized field effect transistors, which was my PhD work. And in this case, we wanted to miniaturize these sensors to become neural probes that we can actually implant in vivo. And then I'll also talk about these Aptamer-Modified Nanopipettes that are for the in vitro sensing applications that I'm now working on at the Laboratory of Biosensors and Bioelectronics. And so I'll have two stories, two slightly different applications, but with the same goal of small molecule neurochemical detection. And so field effect transistors are extremely advantageous because they're fast, they're ultra sensitive, and they're label-free techniques. And to briefly walk you through how they work, you have these source and drain metal electrodes. In our case, they were made of gold. And then you have the semiconducting channel. In our case, it was indium oxide, and it was a thin film of approximately 4 nanometers. Then we assemble the recognition elements or aptamers on the surface of that indium oxide. And then these molecules gate the underlying channels. And what I mean by that is it alters the way in which the current flows from the source to the drain as a function of target capture because they undergo the conformational rearrangement. But one of the disadvantages of working with field effect transistors in high ionic environments is the Debye length. And so if we wanted to conduct these measurements in vivo ultimately, this is a high ionic condition environment. And the Debye length, or effective ion screening distance, is related to the ionic strength of the solution. So if we take a look at a typical phosphate buffer that is undiluted, the Debye length is less than a nanometer. What that means is that anything further than a nanometer from the surface of the semiconductor is very difficult to detect because of the ionic screening. And that's why in a lot of papers, there have been research done in diluted environments, such as 10-fold dilution and 100-fold dilution, because then that sensing regime is significantly improved. But what I can show you here is another advantage of the aptamers, because if we take a look at enzyme-based field effect transistors or antibody-based field effect transistors, the recognition event of the target binding happens significantly further than that Debye screening length. However, since aptamers are smaller in size as well, they can actually be within that Debye length. And further, because we wanted these conformationally changing aptamers, now we can actually detect these changes in the negatively charged backbone in close proximity to the surface of the transistors. And so with that in mind, we isolated in collaboration with Columbia University, the first serotonin aptamer and also a new dopamine aptamer. And so these were conformationally changing aptamers that were isolated through a selection procedure called SELEX. And in doing so, we were able to show that these aptamers could detect serotonin and dopamine in a very specific manner in 1x, so undiluted artificial cerebrospinal fluid. So this is an environment that mimics the in vivo brain microenvironment in terms of the ionic content. And as you can see, we were able to detect very low concentrations in the femtomolar regime. And also we have the perfect control molecule, because basically we design these scrambled sequences where we take the same number of nucleotides as the correct sequence. We also retain the ratio of bases, but we scramble up the order. We model the secondary structure to ensure that it's a very different secondary structure from the correct sequence. As you can see in the presence of that sequence, we don't see that specific response, demonstrating sequence specificity. We also demonstrate that these neurotransmitter aptamers are selective. And so these are precursors and metabolites that I previously showed you in the biosynthetic pathway of these two molecules. And these aptamers can differentiate these with high selectivity. And so something else that we tried was detecting serotonin ex vivo in brain tissue. So in this case, we had the brain tissue from these special mice. They were TPH2 knockout mice. TPH2 is tryptophan hydroxylase 2. It's the rate-limiting enzyme for serotonin biosynthesis. And so these mice basically lack serotonin in their central nervous system. What this allowed us to do was to be able to put brain tissue on our sensors, but then sequentially spike known concentrations of serotonin to demonstrate that we can have serotonin-specific responses in brain tissue. We show that we can sense in a physiologically relevant range from 1 picomolar to 10 nanomolar. That's the shaded area in yellow. And this is based on reported concentration levels of serotonin in the extracellular space. And we also demonstrate that we can reproducibly measure and create this calibration curve for up to 4 hours in that brain tissue, which shows minimal fouling. And so from here, something else that we really wanted to understand was how are we actually sensing? We know that we can sense serotonin and dopamine, 2 small molecules with high specificity and selectivity, but how is this happening? And so to do that, we took a look at the transfer curves. This is the current versus voltage curve for the transistor. And what was very interesting was that for the dopamine aptamer, if you have this dotted line, which is the baseline, with increasing concentrations of the target, we saw a decrease until we reach saturation because all of the receptors are occupied. So from this, we hypothesized that maybe it's because the DNA backbone is moving closer to the semiconducting surface upon target capture. This causes electrostatic repulsion of the surface, creating electron depletion at the indium oxide semiconductor, and therefore you see a decrease in current that goes from the source to the drain. Alternatively, for serotonin, it's possible that from the baseline, we see an increase in current until saturation because of the fact that we see the opposite behavior. The backbone moves away from the surface, therefore you get charge accumulation in close proximity to the semiconducting surface, and therefore you see an increase in current. And so because we had this hypothesis based upon the secondary structure of the DNA upon target capture, we wanted to test this through alternative means. So we conducted circular dichroism spectroscopy. And so this is a method that allows you to see the secondary structure of biomolecules based on tracking the dipole moments and the base stack inside of the sequence. And as you can see for the dopamine aptamer, the dotted line is the aptamer by itself. And upon target incubation, you see a shift in the spectra to the left. And actually the location and wavelength of these peak shifts is indicative of a parallel G quadruplex. This is very established in the literature. In the case of the serotonin aptamer, we go from the dotted line, which is the aptamer by itself, and then we shift to the right upon target addition, and this now corresponds to an anti-parallel G quadruplex formation. And so as you can see, the fact that we're forming 2 new structures based upon target capture also led us to think that the Aptamer conformational change is a really huge factor in the way in which we're detecting these molecules. What's also really nice about this technique is that it's a way to show the selectivity, because we show that in the presence of the non-specific targets, we don't see a shift in the spectra like we did for the specific sequences. And so this also shows that the inherent selectivity likely comes from the fact that in the presence of the wrong molecule, aptamers don't undergo conformational change, at least the ones that we were working with. What is more, we wanted to show that the signal transduction is driven by the conformational change primarily, rather than the target capture, because serotonin and dopamine have a single positive charge at physiological pH. And so we wanted to show that we can even sense neutral targets. And so we isolated the first glucose aptamer, and in this case, we test against fructose and galactose, which both have very similar structures to the target of interest. And also we test, of course, with the scrambled sequence and show that we have glucose-specific responses. And in this work, we also sensed glucose in diluted whole blood. And the fact that we could detect glucose, which is a neutral molecule, really led us to think that the Aptamer conformational change is a really key factor. And so this work, by the time I was leaving my PhD, was going in towards the miniaturization process. And so what we envisioned were these aptamer FETs on the surface of microprobes that are approximately 120 microns in width. And then you can have multiple active areas, you can have inactive areas, for example, with the control scrambled sequence, so that you can always be able to detect the specific binding events. And from the last time I left, I saw that we were already fabricating these devices. We had some preliminary measurements that were highly promising. So I'm really excited what's going to come in the next, hopefully, year or so from UCLA for my coworkers who have been continuing to work on this. But now switching gears a little bit, I wanted to now talk about the in vitro aspect. And so this is now my postdoc work, where now understanding how those Aptamer functionalized field effect transistors were working, what I wanted to try was if we were able to functionalize the inside of these nanopipettes, would we be able to monitor neurochemical flux that is being released from patterned neuronal networks? And so in this case, as you can see, we have the silver chloride reference electrode in the bulk, and also one inside of the nanopipette. And basically we're monitoring the ionic flux that goes through this pore opening as a function of how the aptamers are behaving inside. And so to show you how we actually do these measurements, we measure a current versus voltage behavior. And I'm sure a lot of people in the audience who have worked with pipettes are aware of the ion current rectification behavior, where basically depending on the geometry of the pipette, in this case, it's a conical shape. And we also have a very small pore size of approximately 10 nanometers, which is very similar to the Debye length inside of the pore. We see this nonlinear behavior in the current response upon applying linear voltage. And this ion current rectification behavior has been really well studied for these nanopipettes. And we thought that this would be a way for us to monitor ionic flux change based upon target capture. And what's really nice is that we have this pipette puller in our lab that allows us to create these nanopipettes in a very high throughput way. And so we take a glass capillary, we can heat the center of the capillary with a carbon dioxide laser. By altering the heat intensity and the heat duration, we can alter the pore size of these pipettes. And the ones that we have been working with, as you can see from this transmission electron microscopy image, have approximately 10 nanometer opening sizes. These were optimized for our specific system because we wanted the pore size to approach the size of the aptamers that were undergoing the conformational changes. And so what we wanted to try was, okay, if we were able to functionalize the inside of these nanopipettes, first, we would get some kind of ion current rectification behavior based upon the surface charge at the surface, based on the ionic flux through this small pore opening, and based on the geometry of the pipette. However, upon target addition, there's going to be this conformational change, and will we be able to see a change in the current response as a function of this? And so the first step was to be able to actually put the aptamers inside of these nanopipettes. And so surface chemistry is my background from my PhD. So this was actually the fun part. And you take a quartz pipette, which has these hydroxyl groups on the surfaces, but then we deposit aminosilanes through vapor phase deposition, which actually gives you a nice monolayer compared to solution-based methods. And then we couple the aptamers that are thiolated to these aminosilane groups through malato-benzoic acid coupling chemistry. And then these pipettes are ready to conduct sensing measurements. And what I really like about these nanopipette systems is the quality control. You can actually track this surface chemistry and validate that what you're actually seeing is correct. And so if you start out with a bare quartz pipette, we have slightly negative surface because of the hydroxyl groups. But as you can see, the current response is almost linear. It's not very rectified to start. And then when we assemble the aminosilanes, these amines are now positively charged. And so we get this inversion of the rectification, and we see that there's a change at the surface. Finally, when we add the aptamers, these are highly negatively charged molecules. There's 40 negative charge per DNA, and you see this new rectification behavior where it's a highly negatively charged surface. We can also quantify this rectification using the rectification coefficient, where this is basically just the natural log of the current at a certain potential over same magnitude potential, but at negative bias. And as you can see, the bare quartz has a slightly negative charge, aminosilanes gives you a positive charge, and finally, the serotonin aptamer gives you the highest negative charge, and everything is statistically significant. And so now that we validated that we can actually functionalize these pipettes, we demonstrate that we can sense serotonin specifically in complex environments. And what I mean by complex environment is that these in vitro neuronal networks have to live in this neurobasal medium. Neurobasal medium consists of 17 amino acids, a bunch of growth factors, proteins, other things that keep the neurons happy to survive. And so we had to be able to sense serotonin specifically in the presence of all of these interfering molecules. What we show here is that we see a concentration-specific response with the serotonin nanopipettes, and also that we're able to sense very low concentration in the picomolar regime. And of course, the scrambled sequence does not show a specific response, demonstrating that this is a sequence-specific phenomenon. We also demonstrate that they are selective. One of the amino acids in the neurobasal medium is actually L-tryptophan, which is the serotonin precursor, but we also tested other molecules such as L5-hydroxytryptophan and 5-HIAA to also show that we can differentiate similarly structured molecules. As you can see from this real-time curve, when we inject in the L5-HTP, there's not much change in the baseline current. Same for the 5-HIAA, and with serotonin, we see this increase in the current. And we can show that we can statistically relevantly differentiate these neurochemicals. And so it was really exciting for us to show that we can sense in this complex media, in addition to, of course, simpler media such as phosphate buffered saline. But now we wanted to go and actually sense from serotonergic neurons. The challenge being the serotonergic neurons reside in the raphe nuclei. This is deep within the brainstem. So they're highly inaccessible, and also serotonergic neurons are about less than 1% of the total neurons in your brain. And so to tackle this challenge, actually, our now collaborators at the Salk Institute have developed serotonergic neurons from induced pluripotent stem cells. And so in this case, they start with fibroblasts that are skin cells, and then they reprogram them into these iPSCs. And then finally, they can be differentiated into serotonin-specific neurons. And so this collaboration was really fruitful because basically they were able to send us neurobasal media in which these serotonergic neurons were cultured. And also they treated these serotonergic neurons with different drugs and different treatments to show that we could differentiate these physiological conditions. And so what we show here is that if we have non-serotonergic cells in the media, in this case, they were astrocytes, these are helper cells that are not neurons. And these we were unable to detect in the solution of the neurobasal media in which they were cultured. But on the other hand, for serotonergic neurons, you can see that we're able to detect a change in the current response from the baseline. If we take a look at this simplified diagram for a serotonergic neuron that is communicating, you can see that the neurotransmitter is released, and it can either get bound to the receptor of the next neuron, or it can be reuptaken by a serotonin transporter protein. And so one of the different conditions that we tried was blocking this reuptake using an antidepressant. And so Krishna and her coworkers worked with citalopram, which is a commonly prescribed antidepressant, and it's known to block the reuptake of the serotonin transport protein, and therefore you get an increase in the concentration of serotonin in the extracellular space. And that is something that we were able to detect with our nanopipettes. Further, they also induced neuron depolarization with high salt, potassium chloride. So in this case, all the vesicles that were inside the original neuron gets dumped out into the extracellular space. You get a much higher concentration of serotonin, and that's something that we are also able to detect with statistical significance. And so this is really exciting. And so what we really want to move on to is to conduct neurochemical flux mapping in real time. In our lab, we have the scanning ion tunneling microscopy technique that Dmitry Mamotenko, who is at the talk today, brought to LBB. And this opportunity is really cool because we can actually put these probes inside of this technique and actually map the flux of neurochemicals in real time. But that would require either getting these serotonergic neurons from the Salk Institute and trying to pattern them ourselves in the lab, or alternatively, we can go after a dopamine aptamer, which we've already isolated in our previous work. And what we need to do is to optimize these nanopipettes to be able to gate the opening and closing of the ionic flux based upon the dopamine conformational change. And that's something we're working on right now because we know that the neurons that we have in the lab release dopamine much more than serotonin. But something that I also wanted to talk about in addition to this ultimate goal of mapping the neurochemicals is what we wanted to do in parallel, which was understanding the magnitude of conformational change that's happening inside of these pores. And so a technique that we have in the lab is quartz crystal microbalance with dissipation monitoring. And basically we have this quartz crystal that reverberates at a certain resonance frequency upon applying an oscillatory voltage. And as you can see, basically the frequency, this is the blue line, decreases when you start assembling molecules on the surface, because then you have mass at the surface and the frequency decreases. And again, if you add now antibodies to the surface, for example, you get a further decrease in the frequency because now you have a thicker layer. And we can also monitor the dissipation behavior. And what this means is that when you stop the oscillatory voltage, it's how long it takes for the crystal to stop vibrating. And that actually gives you the characteristics such as the softness of the film, all the different surface characteristics. And so what we did was we tracked the aptamer assembly on the surface of silicon dioxide chips using the same chemistry as the inside of the nanopipette. And so when we inject the aptamer, you can see that we see a decrease in the frequency because now we're assembling something on the surface. And then when we add the serotonin target, we also further see a decrease. Now using viscoelastic modeling, what we can show is that this can be related to the change in the thickness at the surface of the crystal. And so first, when we assemble the aptamer, there is a change of approximately 5.5 nanometers assembled on the surface. And then when we assemble, well, when we add the target and we undergo this conformational change, we see a change of approximately 1.2 nanometers, so a much smaller change. And now if we zoom in on this part, we can show you that upon serotonin target incubation, we see a decrease in the frequency, which is indicative of the aptamer layer increasing in width. And then upon injecting phosphate buffer, we can reverse this binding behavior because aptamers bind and unbind based upon their affinity constants or KD values. And so we also test with a scrambled sequence and show that we don't see this kind of binding and unbinding behavior when you don't assemble the correct sequence at the surface. And what was nice was that the aptamer layer thickness change is something that we predicted previously with the field effect transistors as well, of the serotonin aptamer moving away from the surface, leading to an increase in the current response. Another technique that we worked with is electrochemical impedance spectroscopy. So in this case, we wanted to see how well a redox molecule, in this case ferrocyanide, can access the surface based upon the conformation of the DNA at the surface. And so first, if we have the DNA at some conformation, the ferrocyanide can maybe not access the surface as well, but then in the presence of the target, the aptamer undergoes this change, and now the accessibility may change. And we do indeed see this from the Nyquist plots. And what you see here is that there's a decrease in the width or the diameter of these semicircles. And this shift to the left, or a smaller semicircle, basically shows that there's a smaller charge transfer resistance, meaning that it's easier for these ferrocyanide molecules to access the surface. And so again, this led us to believe that the way in which the aptamer was changing based on all of these complementary techniques was in the same direction as what we were previously seeing. And so this mechanism of the aptamer folding and unfolding inside of these pores is what's allowing us to gate the flux of the ions. And understanding these mechanisms is really important because it really enables us to integrate these aptamers into new systems. And so in this case, it was inside of these nanopipettes. For my PhD, it was on the surface of transistors, but I wanted to show you a little sneak preview of some other things that we've been working on. And so, for example, the microelectrode arrays in which we pattern the neuronal networks directly, I can actually functionalize those electrodes with the aptamers as well. And so in this case, the surface chemistry is slightly different, but in this case, we just directly assemble the thiolated aptamers on gold microelectrodes. And so the nanopipette comes from the top to measure the neurochemical flux, whereas in this case, we're going from the bottom to measure the neurochemical flux. From our preliminary data, what we can see is that if we assemble serotonin aptamers on the surface of these electrodes, we see a change in the surface potential when you add serotonin to the surface. When you have the wrong aptamer, you don't see this. And what's also interesting is that the binding characteristic in terms of the temporal resolution is very different from what we see for the electrical responses. For neuronal firing, these spikes are on the order of milliseconds, whereas the binding behavior is on the order of seconds. So this gives us the confidence that we'll be able to differentiate electrical and chemical signals that are happening at the same electrodes. We also demonstrate that this is a target-specific response at the surface of these microelectrode arrays by testing with 5-HIAA. However, since we were just using the conventional microelectrode arrays that we just commercially buy, we were only able to add targets more into that container, we weren't able to take out that the neurotransmitters that we already added, and therefore we also integrated microfluidics to be able to add chemicals, take out chemicals, do rinsing steps, and as you can see now, we can do measurements such as adding serotonin, seeing a change in the surface potential, and then switching to phosphate buffer and showing the reversible behavior. And so this will also allow us to look more into the aptamer kinetics, and this is ongoing work in the group as well. And so at this point, I'd like to acknowledge everyone that's been involved in this work. Of course, I need to mention my supervisors from UCLA for all the work that I talked about at the beginning. And I also want to thank the Laboratory of Biosensors and Bioelectronics, specifically Dimitri, who's been working on this work with me for the nanopipettes all these years. And I'd like to, of course, thank the sponsors such as the FNSNF, the Graphene Flagship Project. Alex here is one of the students who worked with me on the Nanopipette project, as well as Dominic. I'm very grateful to all the students who have contributed some great work towards this project as well. And finally, the Salk Institute collaborators, Krishna and Randy Gage, who also enabled us to measure in real complex environments, which is really exciting. And so at this point, I'm very happy to answer any of your questions, and thank you very much for the opportunity and for your attention.
Thanks so much, Nako. That was incredibly interdisciplinary work, quite spectacular, I would say. So I'm hoping that there'll be lots of questions, and maybe if you can just stop sharing your screen.